So thank you for joining us today for brain health and blood sugar. We would like to note that the information in this presentation is for educational purposes only and does not constitute medical advice. Our information is intended to empower individuals, their families, and healthcare professionals who want to collaborate in the most effective ways on this journey to health and well-being. Please see a licensed and qualified medical professional for your medical needs. If you have any questions during the presentation, please type them in the chat box, and we will answer those during the Q&A at the end of the presentation. Before I begin, I would like to give a short program update, and then I will introduce our speaker. Corey, if you could advance that slide. Thank you. Here at Sharp Again, our mission is to educate and empower people to take charge of their cognitive health. We do that by helping to support people through webinars like this one, in-person presentations, and our printed materials, along with our informative website. But by far, the most effective offering is our small group coaching program. This program offers live education and support facilitated by a certified health coach. Now going into our fourth year, we are pleased to let you know we have a program coming up. It begins on April 25th and meets via Zoom once a week for 6 consecutive weeks. In the small group program, participants begin to identify and modify lifestyle factors that might be impacting their brain health. They learn from one another's experiences, and this community experience has been the highest rated aspect of the program. During this small group program, we focus on four lifestyle factors that are known to contribute to cognitive issues: diet, nutrition, exercise, stress, and sleep. Participants have access to an online teaching platform for the educational videos, quizzes, and resources that accompany each topic module, and that prepare them for the virtual coaching sessions. The price of this program is only $99, and it is significantly subsidized through the generosity of our Sharp Again donors. The actual cost of the program is $450 per person, so it is an enormous value. If you're just beginning to notice some memory impairment, or you want to learn how to keep your brain sharp, this program is an excellent place to start. In addition to this program, we also offer our Maintain Your Brain community, which offers continuing support for our foundational group graduates. It meets twice monthly to help people stay on track with the lifestyle changes they are making to maintain their brain health. On the next slide, we have information about our upcoming webinar. On Tuesday, April 9th at 12 p.m. Eastern Time, Robert Grable, who is a Sharp Again board member, will be joining us for Finding Light in the Darkness, a Dementia Journey. Robert's mother was diagnosed with vascular dementia 5 years ago, and he has learned vital lessons through his relationship with her and in caring for her over those years. He will join us to offer actions we can take immediately to enjoy and support our loved ones. Now, it gives me great pleasure to introduce our speaker, Dr. Cornelia Lenherr. Dr. Lenherr is a family medicine physician with Community Health Programs Health Center in Great Barrington, Massachusetts, and she is a member of the Sharp Again Medical and Dental Advisory Board. She received her bachelor's in pre-medical studies at Simon's Rock College of Bard and earned her medical degree at Dartmouth Medical School. She completed her family medicine residency at the University of Rochester Highland Hospital and also completed a fellowship in family systems medicine at Highland Hospital with a focus on family therapy training. Her approach to care incorporates her training in muscle release therapy, psychoanalysis, stress reduction, mind body medicine, meditation, biofeedback, and other health management techniques. She is a board certified family medicine physician and a certified functional medicine specialist. Welcome, Dr. Lenherr. Thank you so much for joining us.
Before we get started, I'm going to run through a few abbreviations. It makes the slides a little easier if I don't have to type out all these long words every time, but at the same time, the abbreviations can be befuddling. So 2TDM is for type 2 diabetes mellitus, which is the adult onset version of diabetes. IR is insulin resistance. AD Alzheimer's, FBG is fasting blood glucose, BG is blood glucose, HTN hypertension, and hepatic glucose production is HGP. There will be a quiz. So going forward here, we're here because of the significant burden of dementia. Globally, more than 55 million people are suffering with dementia and approximately 10 million cases every year, new cases every year. As many of you know, there are different kinds of dementia. It's an umbrella term. And so conditions like Alzheimer's, Lewy body, frontal temporal lobe dementia, vascular, and Parkinson related all fall under the umbrella of dementia. But they have slightly different characteristics. What they have in common is that they're ultimately conditions or diseases that involve neurodegeneration. Dementia affects cognitive functions such as memory, thinking, calculation, learning, language, and so on. This is a slide that shows the risk factors related to dementia, and many of these you may have heard of. I'm going to highlight a few here. So smoking, high blood pressure, physical inactivity, and diabetes are all very common habits, conditions in our culture, in our world, and they can play a role in the development of dementia. So hyperglycemia is well documented as being related to the genesis of neurodegenerative diseases. Hyperglycemia is a major risk factor for cognitive impairment and dementia. Insulin resistance is related to Alzheimer's disease or linked with Alzheimer's disease. Those who have type 2 diabetes are 1.5 to 2.5 times more likely to have neurologic complications. And that involves things beyond dementia. Oftentimes, people with type 2 diabetes have things like neuropathy or changes in sensation in their feet and other parts of their bodies. Alzheimer's is more likely or more common in diabetic patients, and poor glycemic control is related to higher risks of cognitive impairment. Don't worry about these special terms. I'm going to be explaining them all as we go along, okay? So as a family physician, I work with diabetes and high blood sugar and pre-diabetes all the time, every day. Less commonly, I'm dealing with neurocognitive decline. But as a physician, when I do labs and get a blood sugar on somebody and it comes back not so pretty, the kinds of comments and discussions that I have are pretty common. And I mean, the comments are pretty characteristic. So things like, I don't need any sugar. How can it be high? How do I know when my blood sugar is too high? Are carbohydrates bad? And these questions that I've listed here on this page are really typical of what people are thinking about and how they respond when we're talking about an abnormal blood sugar. During the course of this talk, I will answer all of these questions. So first, we're going to do a little bit of dive or detour into understanding blood sugar and carbohydrate chemistry. So sugar is the common term for carbohydrates. It's not very specific. It's not scientific at all, and but it's often what we use when we're talking about carbohydrates. Blood sugar is the common term for blood glucose. So glucose is a sugar, but there's more than one type of sugar that can end up in the bloodstream and does end up in the bloodstream. So I have a couple little pictures to share. The graphic, the diagram on the left is a glucose molecule. And as you can see, it's a ring shape. It's a has 6 atoms in it, 5 carbons and an oxygen. And 4 of the carbons are attached an HOH moiety, as it's called in chemistry. And that is actually H2O, which is water. And so when you take a carbon and you hydrate it with water, it becomes a carbohydrate, which is the little graphic on the right. Now, going forward with naming sugars and sugar nomenclature, the Greek term for sugar is saccharide. And probably many of you have heard of things like monosaccharide, disaccharide, oligosaccharide. And what that's denoting is the number of sugar molecules in the element. So sugars with one that are single molecule, mono, are have one molecule, and some common examples are glucose and fructose. A disaccharide is a joining of 2 molecules, and ones that you may recognize are sucrose and lactose. Lactose is milk sugar. And then oligo is 3 to 10, and those are things like cellulose and starch. Generally, oligosaccharides are chains of glucose alone, but not exclusively. Now, sugar, the table sugar, the white stuff, its chemical name is sucrose. And sucrose is a combination of fructose and glucose. It is broken down in the small intestine by an enzyme known as a disaccharidase. And the graphic on the right shows a glucose molecule, which is the part on the left, and a fructose molecule, which is a little bit different because it's a 5-member ring versus a 6. And fructose is the sweet part of glucose. I don't know if any of you are aware, but glucose is not very sweet. And something like Karo syrup, which is a glucose syrup, is not a very sweet product. So when the disaccharide is broken down, the glucose ultimately enters the bloodstream, and then our blood sugar rises. Now, glucose is good, okay? It is cellular fuel. It's essential for life. And as a cellular fuel, it's essential for the generation of ATP, which stands for adenosine triphosphate, and that is a basic unit of energy. Our cells are totally dependent. We are dependent on ATP and ATP production. And as an aside here, I just want to say all of this happens in a small unit within the cell called the mitochondria. And I'll make references to mitochondrial function later. So it's a subcellular body within the cells, and it's where all of this ATP generation occurs. So glucose mainly comes from food, specifically carbohydrates. So again, all carbohydrates are made up of sugars. Now they can also contain fiber, and many do, like in our vegetables and fruits and things, but they're essentially connections of glucose and fructose molecules. The liver also synthesizes glucose, and that's the hepatic glucose production. So we have 2 sources of glucose in our bodies. From the pancreas makes insulin, which is a hormone that helps the cells take up glucose. And the specific cell in the pancreas is the beta cell. And muscles, fat, and liver need insulin for the uptake of glucose. So again, one of the questions I get, well, are carbohydrates bad? Should I avoid them? We even have terms for good carbohydrate and bad carbohydrate. But the bottom line is that they are really essential for life. So with all of this, now that you're well-versed in carbohydrate chemistry, we're going to move on to hyperglycemia. And hyperglycemia is really when the blood sugar is high enough, it's at a level that's above a standard reference range, normal reference range, that is. And this reference range can vary based on the timing of the last meal. So we're going to focus on this little glucose chart here. And if you look at the green bar where it says normal on the left, a fasting blood sugar is typically between 80 and 100. Sometimes it can be a little bit lower. Sometimes it can be a point or two higher, and it's still a normal blood sugar. After eating, the blood sugar rises. It can go up to 170, 200-ish, depends on what we eat. But with the rise of glucose is also the secretion of insulin, and insulin then acts on the tissues, which allows the cells to take up glucose from the bloodstream and ultimately leads to a drop of blood sugar. And that's what you see in the fourth column there, where 2 to 3 hours after eating, blood sugars are almost back to the fasting level. So hopefully that makes sense to everyone, and just send me a question if you're not clear on that. Now, what happens if you fall outside of those reference ranges? Well, then what we're moving to the next level, impaired glucose. And you start to have higher than normal blood sugars when you're after fasting and after eating, and then they don't come down to quite the same low level after 2 to 3 hours. This is the category where insulin resistance starts to happen, and where, depending on the level of the blood sugar, we move into the category of having prediabetes. If our blood sugars come back at a level of 126 or higher, then we're in the diabetic range. And as you can see, the levels after eating and 2 to 3 hours after eating are substantially higher than the normal. Now, this having a single blood sugar that's higher than 126 is one of the ways we diagnose diabetes. But we also, by definition, need to have another confirmatory test. And often we use this test called the hemoglobin A1C. I'm sure everyone listening has had it done, and some of you may be very knowledgeable about your own A1Cs. And it's a good test for a number of different reasons. So what an A1C is the average blood sugar over the past 3 months. It represents what your blood sugar has been doing over the last 3 months. It is a glycosylated hemoglobin measure, and essentially it's a sugar-coated protein. And hemoglobin is in the red blood cells. Red blood cells last about 120 days, so a little longer than 3 months. So what we're looking at is how sugary did those hemoglobin molecules get? Good thing about the hemoglobin A1C, it's not dependent on fasting, so we can do them in the office at any time. I'm not sure if there's a over-the-counter version available, but it seems like there should be. And the higher the A1C is, it has a predictive value of being able to predict vascular complications. So let me briefly explain the chart to the left. A normal hemoglobin A1C is from somewhere about 4.5 to 5.7. And you can see it represents a average daily glucose of about 117 or less than 117. Now, again, you're going to eat a meal, your level's going to go way above 117, but on average, it's not going above that over a 3-month period. Prediabetes is when you start to move into the higher ranges of the A1C up to 6.4. And then beyond 6.4 is diabetes. And as you can see, the complications start to really increase when the hemoglobin A1C is beyond 7. All right, so hemoglobin A1C, elevated blood sugar, is technically impaired glucose metabolism. It's all a spectrum. You go from normal to mildly elevated glucose to prediabetes, insulin resistance, and then eventually diabetes, all on a spectrum. And so this is the territory where we have control. We have some ability to intervene and turn around the process of going from mildly elevated to diabetes. The burden of diabetes in the United States is significant. More than a third of the adults in the US have diabetes. Most of it's type 2. There are other types. There's 1.5, type 1, which is when you don't produce any insulin, and it's usually associated with a childhood form of diabetes. The risk for diabetes, in short, are genetics, lifestyle, and aging. And the hallmarks of impaired glucose metabolism are elevated blood sugar, reduced insulin sensitivity at the tissue level, and decreased insulin production. I'm using these terms: impaired glucose metabolism, insulin resistance, type 2 diabetes. They're not identical, but they're basically representing the same principle. The blood sugar's abnormal, and usually some sort of metabolic and whole body abnormalities are underway. So insulin resistance and type 2 diabetes really is defined by an imbalance of plasma glucose and the ability of the cells to use and uptake the glucose. Hyperglycemia and insulin resistance leads to increased glucose production in the liver. And also, paradoxically, paradoxically, it leads to increased glucose production. So, and then there's also decreased glucose uptake by the muscles, liver, and adipose tissue. Aging also has an impact on insulin resistance hyperglycemia, and it does so because of the reduced beta cell proliferation and function in the pancreas. So let me say a little bit more about that. The beta cells is where the insulin is created in the pancreas, and there is regular turnover. So if the cells aren't turning over and functioning properly, then insulin production will go down. Aging also indirectly affects insulin sensitivity via lifestyle choices, and in turn, insulin resistance increases beta cell dysfunction. The next page, I have a graphic of just what I said. If you look at the upper left-hand corner, you see aging, and you go across the top, and it directly affects beta cell function and then diabetes. If you drop down from aging, there's an indirect connection via lifestyle, genetics, other comorbidities, and then that leads to impaired impaired beta cell function, insulin resistance, and ultimately type 2 diabetes. This is another graphic describing the inputs of hyperglycemia. So we can have hyperglycemia because of what we have in our diet and our body's inability to take it out of the bloodstream. We can have elevated blood sugar because the liver makes glucose, and that adds to the burden of hyperglycemia. So the liver schematic, I wish I had a pointer, but I don't. Liver schematic is directly to the right of the word hyperglycemia is a graphic of the liver, and the word gluconeogenesis means to make new sugars. Also below that is a picture of the muscle, and with reduced glucose uptake, there is a process of making more sugar. And some of this has to do with the fact that, in a sense, the tissues are in a relative glucose starvation state. They're not able to get glucose into the cells. The cells are saying, hey, we need glucose. And so they turn on mechanisms that allow for intracellular production of glucose. But that also adds to the blood sugar burden. Moving forward, we're going to take a quick look at some of the effects of hyperglycemia on the brain. Now, this is a dense topic. And so I've done my best to try to pare it down to manageable bits of information. Now, it's clear that dementia risk is increased with insulin resistance and diabetes. That is very well documented in the literature. And back in 1989 was when insulin resistance as a feature of Alzheimer's was first proposed and identified. Clearly, there are a lot of commonalities between the development of Alzheimer's and type 2 diabetes. And what I mean by that are many of the cell signaling processes and biochemical processes are common in type 2 diabetes and Alzheimer's. And so Alzheimer's is sometimes referred to as type 3 diabetes. Now, I've mentioned that they have common pathophysiologic characteristics, and the signaling pathways are also similar. So signaling is quite a complex set of messages that happen between insulin, the cell, the inside of the cell, and the wall of the cell, which opens up a receptor to allow glucose to enter into the cell. And I think it's probably ever evolving in terms of how it's defined and what we know about it. But it's the important way that insulin impacts cell health and gets glucose into the cells. Hyperglycemia is clearly identified with neuroinflammation, so inflammation of brain and neurogenic nerve tissue. It increases oxidative stress, which is basically when you have dangerous ions in the cell and bloodstream, which cause damage to mitochondria, cells, tissue, DNA, etc. There's mitochondrial dysfunction, and all of these are common to both. Now, I just thought I would impress you with a really difficult, busy slide. But at the header here, type 2 diabetes risk factors are listed. So on the left are things like ethnicity, family history, genetic predisposition, and on the right are things like obesity, physical activity, unhealthy diet. All of the down arrows, the big bold down arrows, are pointing to areas of dysfunction. So the orange is pointing to mitochondrial dysfunction, the green to changes in gut health, and the purple to inflammation. Ultimately, these feed through the system, the cells, and the subcellular units. And way at the bottom, there's problems with insulin secretion, beta cell mass, increased inflammation, and DNA damage. So those are the takeaways from this busy slide. Now, further, more on hyperglycemia and brain health. Insulin receptors in the brain, they regulate energy consumption, food intake, behavior, and vascular function. At one time, we didn't know if there were insulin receptors in the brain, and it's very clear that there are. And disrupted insulin signaling alters the glucose uptake by the cells. And I mentioned the signaling earlier, and this is the kind of like a cascade mechanism that helps get glucose into the cell. When the signaling is altered, then that impacts mitochondrial function. And if it's not functioning, the mitochondria aren't functioning well, then there's a feedback of reduced glucose transport into the cell and the formation of tau protein, and then ultimately neurodegeneration. And for those of you who don't know, the tau protein is one of the key hallmarks of Alzheimer's. Now, this is a really super simplified version of what's a complex interplay of intracellular and extracellular factors in type 2 diabetes and Alzheimer's. But essentially, it's altered glucose metabolism, affects mitochondrial function, and then all heck breaks loose. And what is interesting about this research is that the use of anti-diabetic drugs used for diabetes are showing some promise in reducing the risk for dementia in type 2 diabetes. So I have another fun slide for you. And the point of this, again, is to show that this is a really complex set of mechanisms. So at the top, we have insulin resistance. And you can see, similar to the type 2 diabetes slide, there's mitochondrial dysfunction, oxidative stress, brain inflammation. And then again, all these down arrows are impacting different aspects of intracellular and extracellular function. And over on the far left at the bottom, you see mitochondrial dysfunction is highlighted there. If you move along to the right at the bottom there's the uptake of glucose by the brain is impacted, tau protein aggregates, and then ultimately there can be signs of cognitive impairment and ultimately dementia. Now, we've looked at some of the pathophysiology around type 2 diabetes, the connection between hyperglycemia and brain health, and some of the pathology there. So what can we do? What helps this? And so some of the key takeaways from the reading that I did in preparation for this is that exercise is key. Age is an issue, so don't get old. Our diet, specifically if it can promote weight loss, is important. And the role of medications is becoming more central in terms of helping to prevent dementia as we treat diabetes. So back to this graphic that we started with at the beginning. Again, risk factors for dementia. Again, high cholesterol, high blood pressure, smoking, physical inactivity, our diet, diabetes, genetics, they are all going to play a role in dementia. And there's a huge overlap with the impact of these features on the development of type 2 diabetes. Obviously, diabetes isn't a risk factor for diabetes, but genetics, diet, physical activity are. So the best approach is really prevention. It's far more effective in having any impact on the development of disease than attempts to reverse disease once it's started. Excuse me. Once people have moderate to advanced dementia, there's a lot more amyloid plaque and tau deposition and more damage, and that's difficult to reverse. The interventions may stall or halt progression, but they're very unlikely to reverse it. And so what we're talking about with prevention of cognitive impairment and dementia is really a primary prevention. And that means before symptoms have developed, we are forestalling the problem. Secondary prevention is when we start to have signs and symptoms of a disorder. So for example, if we exercise and follow a healthy diet, we may be preventing diabetes or hyperglycemia. We may not even realize we're doing that. But if we start to have high blood sugar or prediabetes, then any intervention at that point would be considered secondary. Can be just as effective, but it's a different category. It's important to understand what we can affect, and those tend to be modifiable risk factors. And then in addition, we want to treat known risk factors, such as high blood pressure. And again, for dementia, we want to manage diabetes and hyperlipidemia, which is high cholesterol. So this is a graphic of modifiable versus non-modifiable risk factors. So on the left are things that we can change. We can change how much we move. We can change habits, although they're hard, such as smoking, alcohol intake, sugar intake, our diets. We can work on stress reduction, sleep, and then also treat other conditions that are associated with diabetes. Things that we obviously don't have control over are our age, our gender, genetics, and then comorbidities. Sometimes we have not, we just can't control it. Somebody with cancer, somebody who already has advanced heart disease, things like that are comorbidities, that meaning health problems that are coexisting, and that and they may have an impact on both diabetes or hyperglycemia and neurodegeneration, but we may not have much control over the course of the comorbidity. So I'm going to focus mostly today on exercise because it's just really profound in terms of its help with diabetes and with Alzheimer's. So it's well established that having a low cardiovascular fitness is an independent predictor of all-cause mortality in patients with type 2 diabetes. So what does that mean? So if we're really inactive, then that is going to be a predictor of the consequences related to our diabetes. And those consequences are things like Alzheimer's, things like heart disease, things like kidney disease. Consistent physical activity reduces the risk of developing impaired glucose tolerance, insulin resistance, diabetes, and dementia. Exercise is a first-line recommendation for patients with newly diagnosed type 2 diabetes. And again, regular exercise has been shown to reduce, not increase, reduce hemoglobin A1C levels and to increase brain-derived neurotrophic factor. So neurotrophic factor, again, comes from the brain, and it is a factor that is known, it's a little like a fertilizer. It helps the neurons and other brain cells and can be a potent factor in stalling and reversing cognitive impairment. Another graphic on the exercise of the benefits of exercise. So there's reduced inflammation, reduced cholesterol, blood lipids, improved glycemic control, insulin signaling, and vascular function. And I just want to point out a couple of tissues. So these are the main tissues that are utilizing glucose, and so with exercise, there in the fatty tissue at the top, adipose, there's decreased inflammation and increased insulin sensitivity, and you'll see increased sensitivity repeated for muscle and liver, and it actually is also true with brain, but it's not on this slide. So this study done by Kiron and Sachs is interesting. It took a group of patients, and for 7 days they had a routine of vigorous aerobic exercise, and the adults all had type 2 diabetes. And what they were able to show in this short study or short time period, is that there was improved glycemic control, a drop in plasma insulin, and 45% increase in the insulin-related glucose disposal. So glucose disposal is the uptake of glucose by the cells. And additionally, a suppression of hepatic glucose production. So remember, hypoglycemia is not only what we get from our diet, but it's also what comes from the liver cell production. So exercise, what types, what do we do, how much, etc. They're all good in different ways. So in the same study or the same paper by Kirwan, they discuss aerobic exercise and show that when you take the intervention group who's doing exercise versus the control group, which is sedentary, that aerobic exercise clearly improves glycemic control, insulin sensitivity, and oxidative capacity. So that's the ability of the cell to take in oxygen, put it into the mitochondria, and use it for production of that famous ATP molecule. High intensity interval training may actually be more effective for glucose regulation and insulin resistance than aerobic training. So HIIT is great for those who are in pretty good shape and can handle those higher intensity bouts of exercise. But for the average person who's been sedentary, already has high blood sugar, extra weight, the joints are hurting, you want to start out with a graduated aerobic and resistance exercise programs. The next category of exercise is resistance training, and that's strength training, lifting weights, anything where you're taking some sort of a weight and moving it in space. It has benefits beyond insulin sensitivity in that it improves strength. It can help with mineral density, although not very much, blood pressure, cardiovascular health, and obviously insulin sensitivity. The current recommendations are to exercise for 150 minutes a week, which, over a 5-day period would be about 30 minutes a day. And the exercise should be vigorous to moderate to vigorous. So no strolling through the mall, that doesn't really count. Okay, so let's see where we are here. Diet. Okay, timing's great. So we're coming to nearing the end of the talk here. I have a little bit to say about diet. It's a huge topic and it's so controversial. And everybody has an opinion about what we should eat and what's going to work best. So what we do know is that when you're carrying extra weight and have hyperglycemia, weight loss is helpful in preventing and improving the management of type 2 diabetes. So it's obviously going to bring down blood sugar and lowers your risk for diabetes. Currently, there are, low carbohydrate, low glycemic index diets, Mediterranean, and very low calorie diets are popular. There is really no consensus on the optimal dietary strategy, and especially what's best for people with obesity and diabetes. And the author of one paper I reviewed said, best diet is one that achieves the best adherence based on the patient's dietary preferences, energy needs, and health status. And that makes a lot of sense. If I prescribe a diet that says, eat nothing but walnuts, I'm going to get cross-eyed looks from my patients. So we do have to take into account the realities of what food means to us and what people are going to be able to sustain. Going forward with diet, studies that look at low carbohydrate and ketogenic diet do comment that they are effective in reducing body weight and improving glycemic control in type 2 diabetes. There's a stronger effect the lower the carbohydrate intake is. So therefore, ketogenic diet is going to have a stronger effect than one that's just lower in carbohydrates. There are some issues with this. The ketogenic lower carbohydrate diet, some of the calories that we might be taking, we'd be replacing, complex carbohydrates for more higher fat foods such as cheeses, meats, things like that. And that could have an impact on cardiovascular risk. So the question is to weigh the cardiovascular risk versus the benefit of weight loss and glycemic control. So that, to some extent, going to be a case-by-case decision-making process. And ultimately, a low carbohydrate may not be appropriate for all individuals. My final slide today really has to do with the use of medications. And there are a lot of newer diabetes medications that are very well tolerated and extremely helpful in the treatment of type 2 diabetes. And what's so interesting is that some of these are being looked at for the treatment of prevention and treatment of early dementia. So the newer medications target specific enzymes and receptors, and a couple in particular increase the pancreatic glucose production. Oh, sorry, that's a mistake. They increase the pancreatic insulin production. So sorry. Unfortunately, a lot of these studies that are looking at this effect of the type 2 diabetes medications on dementia are so far being done in animals. So we can't really extrapolate, but promising animal studies tie the reduction of insulin resistance with improvement of brain function and cellular processes. We can see that there's improvement in working memory, and spatial memory deficits are improved. This research is in its infancy, early stages, and I'm sure there will be a lot more coming forward. One more note on this. One of the classes of drugs that's being used for the research on Alzheimer's or dementia and cognitive impairment are the class of drugs called the GLP-1 agonist, and those are the drugs like Ozempic and Wegovy that are so popular for weight loss these days. So when we use them in patients with diabetes, we see some weight loss in those situations, and the lay public has grasped onto this as a new way to lose weight. So in summary, I lost my slide there. What happened there?
I don't know.
There we go. Well, is that good enough? Will that work?
Sure.
Okay.
Oh,
Where's
it
my
looks like maybe
I don't have it. Well, I'll read it. I'm sorry, folks. So in summary, cognitive decline leading to dementia is a complex process which is influenced in part by age, genetics, environmental exposures, metabolic diseases, and lack of exercise. Prevention and reduction in decline is best achieved by maximizing health as a whole. Yeah, I apologize for not having that slide in there. I don't know how that happened. So that concludes my talk for today, and we have a few minutes for questions.
yes. So one of our first questions, what is the difference between hyperglycemia and hypoglycemia, and what are the symptoms?
Great question. I almost had a slide for you, but I didn't include it. So hyper means that your blood sugar is above a standard reference range. So that's that colorful chart that we looked at early on, where, if the fasting level is above 100, it's starting to become a hyperglycemic picture. Not necessarily a problem, but that's how it would be defined. Hypo is when blood sugar drops below a standard reference range. And in this case, it actually depends on the lab, but it would be somewhere between below a reading of 60 to 80. And so somebody who normally has a blood sugar, say, runs around 90, when if you haven't eaten for a few hours, your blood sugar may be hovering around 90 to 100. If you were to have a sudden blood sugar drop, even of 20 points, you would feel it. And you feel lightheaded, grouchy, hungry, lack of ability to think clearly. Hypoglycemia is often related to, it can be reactive. So if you eat a meal that contains a lot of simple sugars, you might get a hypoglycemic response because of the insulin. It's rare as a standalone condition. And I think that's sounds good to me. Next question.
This one kind of moves into medical advice, but you might be able to give some insight into what metformin does. The question is, my A1C is 5.9. My doctor doesn't think I should be on metformin. I am 83, slim, active, and follow a low-carb diet. Why not take metformin? I'm guessing
Okay. So
risking low blood sugar.
Yep. So could you read the A1? What's the A1C again? 5.7?
5.9.
9. Okay. So I'm going to just generalize here, but the A1C being 5.9 is just slightly, it's in the prediabetes range. It would be nice to know fasting blood sugar along with that, so that adds to the clinical picture. But what you've already said is there's not an issue with extra weight. There's, it sounds like good activity and a diet that suits your needs. Metformin increases tissue sensitivity to glucose, and so to insulin, and therefore better uptake of glucose. So it works well in the skeletal tissue, the fat, and the liver. It is not the kind of medication that stimulates the pancreas to put out more insulin. It works on the sensitivity issue on the tissue side. It's usually the first go-to drug in treatment of diabetes, generally well tolerated, although it can cause some gastrointestinal side effects, which may or may not be tolerable for someone. So I can't really answer why not, why take it, why not take it. But it sounds like having more conversation with your physician would be in order.
Another question, you mentioned carbs as the primary source of glucose. What are your thoughts on being fat adapted and gluconeogenesis?
Let's see. Okay, so if you're fat adapted, meaning I'm assuming you mean that you're on a very low carbohydrate diet and are in a state of ketogenesis and using fats for your primary fuel. That is an alternative fuel. It is people, that's part of what people are doing with the low carbohydrate ketogenic diets for health. It doesn't have a lot of long-term studies, so we don't really know. It's too early in time for us to know whether there are significant issues with that. But I will say the ketogenic state is a survival mechanism that we humans are very lucky to have. So if we were totally dependent on glucose to run our cells, then any period of time without food could easily cause starvation and death in a short period of time. And as an example, chimpanzees cannot go into ketogenesis. So they need to keep close to their food source. So the alternative fuel of ketones is probably an evolutionary adaptation that's allowed us to survive at times of low food availability.
And one last question, can insulin levels be tested?
Yes, they can. So it's not commonly done, say in a primary care office. I have done it in the past through my functional medicine practices. Its utility is, so-so. The more readily available tests of the blood sugar, which, people can test their own blood sugars at home, and then the hemoglobin A1C are the most common and universally accepted ways of being able to see how, what's going on with our metabolism. But it, may be helpful to see how high your morning insulins are and see if you, tend to run on the high side.
The other thing I wanted to mention, there are numerous home A1C testing kits available over the counter.
Thank you.
You're welcome. My daughter has type 1 and her endocrinologist, APRN, uses those home tests because she doesn't have a machine in her office.
Okay.
So?
Okay. For A1C and not for glucose.
For A1Cs.
Super. Okay. Yep. Okay.
They're, and they seem to range in prices quite dramatically. I don't know which are good and which are bad.
Yeah.
Well,
So can I just say one little tack on to that? So the thing about the A1C is that since it's a 3-month measure, if you had an A1C of 7 and you said, great, I'm going on an exercise diet program, and you started checking your A1C, it would be a while before there would be a noticeable drop because of the turnover of the red blood cells. So
interesting.
thank you.
Well, thank you so much, Dr. Lenherr. This was
Pleasure.
enlightening and so educational. I will be sending out the link to our YouTube channel of this recording tomorrow, along with information about our upcoming programs. And I'll try and include some links to some information that Dr. Lenherr touched on this afternoon. Okay.
Thank you, everyone.